Today I'd like to give you a short overview of how to use our images to analyze caspase activity in living cells over time. Personally, I'm really excited about this application as it is very simple and suitable for automation and high-throughput screening. To perform this assay, we use two fluorescent probes. One is the CellEvent Caspase-3 Green Detection Reagent. In this reagent, a high-affinity DNA binding dye is linked to a Caspase-3 peptide substrate. It enters cells, but only upon Caspase-3 cleavage, the dye is released, binds to DNA, and fluoresces. We combined this reagent with the probe Spy650 DNA, which is membrane permeable and can thus enter all cells in which it fluoresces upon DNA binding. In our simple add, no wash, no lyse assay, we seeded pancreatic cancer cells on 96-well plates. After 24 hours, we added a staining solution containing the two dyes in medium and a treatment solution containing various concentrations of the chemotherapeutic drug gemcitabine. Half an hour after the addition of the dyes and treatment, we started the first measurement and imaged daily for 72 hours. Here you see an example experiment directly in YT software. In the setup window you can see the layout of this experiment. In column 2 we had the negative control, while in row 3 till 10 the cells were treated with rising concentrations of gemcitabine. In the evaluation window we can select the wells of interest in the Analyst tab. When we look at the last measurement in the control-treated wells, we see an almost confluent monolayer of cells with red Spy650 DNA positive cells. There are also some green cells with caspase activity, as in every cell culture some cells will die. However, most of them look nice and healthy even after 72 hours. You can also select the previous measurements to observe the development over time. Now, if we look into a well in which the cells were treated with a high gemcitabine concentration, we see a lot more cells with caspase activity and some nuclei with bright red condensed or fragmented DNA. Cell shrinkage, membrane blabbing, and extracellular vesicles or debris is visible. However, at the first measurement, the cells look comparable to the control-treated cells. Using our virtual cytoplasm 1F application, we can quantify these results. The Spy650 DNA positive nuclei are encircled in green, and if they are surrounded by a green fluorescent signal, they are marked by an orange circle as shown here. In the Analyst tab, we can look at the heat map for each time point. For example, here we see that the average nuclei count after 72 hours is around 60,000 for the control-treated wells, while it is only 8,000 for the highest gemcitabine concentration. If we switch the property to the percentage of cells with caspase activity, we see around 66% in the gemcitabine-treated wells and only 3.5% in the control wells. We can also select the time chart function to get an overview of the development over time. The results are also visible in a spreadsheet format and can be exported. We calculated dose response curves for the two parameters nuclei count and percentage of cells with caspase activity, as shown here. In our case, we simultaneously imaged the cells on the bright field channel and also analyzed the confluence of the cells. This is detailedly explained in a separate video or the application note, but here you see a short summary of the results. In a last step, we simply added a viability assay and again measured it in I1. Using this high-content screening approach, we could analyze the confluence, nuclei count, and caspase activity in one single plate over time and compare the results with a standard endpoint viability assay. As I said in the beginning, I think it's a very cool and easy assay telling you a lot about your cells and treatments. If you became curious and want to find out more, visit our website at synentec.com or contact us directly via appsupport@synentec.com.